In this session, we'll be discussing about one of the sexual reproduction methods in bacteria, which is called transformation. Now, what happens in transformation? That the transformation experiment, it's been set by this particular scientist called Griffith. And Griffith worked upon Diplococcus pneumoniae. It's a pneumonia-causing bacteria. Somewhere you will feel the name that it has been different, that it is Streptococcus pneumoniae. Streptococcus pneumoniae and Diplococcus pneumoniae are the same bacteria. Now what happens in Diplococcus pneumoniae? He has worked for two strains of this particular bacteria. One is R2 strain, one is S3 strain. Hopefully you know that I have already told you that strain are when the particular bacteria are same with their genus and a specific epithet, but still the bacteria are different, means some minor differences are there. The bacteria will be different at their strain level. So the R2 and S3, they belong to the same genus and same specific epithet, but still they are different, and because of that they have been given with the two different strain. One is R2 strain, another is S3 strain. R stands for rough strain. This particular bacteria, the wall is rough, and because of this roughness, it doesn't carry any toxic protein, and that particular bacteria becomes non-virulent or non-pathogenic. When you talk about S3 bacteria, the bacteria is smooth, and because of the smoothness, bacteria becomes virulent or pathogenic, means it will be causing the pneumonia. Now what happens, what he did is, the Griffith, first of all, he injected R2 strain to the mice. R2, I've already told you, it is non-virulent. So when he injected non-virulent strain to the mice was alive. But what he did is, in the second experiment, he took the strain, S3 strain, and he tried giving the S3 strain to the mice. S3 causes the infection and the mice died. What did he do in the third experiment? That when he thought that, okay, this S3 is killing, he heat killed the S3 strain. When he gave this heat killed S3 strain to the mice, because of this heat killed S3 strain, the toxic proteins were damaged, mice was alive. Now what did he do is, in the first experiment, he took R2 strain where the mice was not dying. In the third experiment, he took heat killed S strain where the mice was also not dying. Now what happened in the fourth experiment? What did he do is, he gave the combination of first and third. And now he want to see that what is happening. So first means rough strain and third means heat killed smooth strain. And what happened to the surprise is mice died, because it was not dying here, but it died here. So he thought he must be running something wrong with the experiment. He tried repeating it for the several times, and again mice was dying. So then he thought that R2 was not killing, S3 heat kill was not killing, then what has been happening? So he told there is something which is transferring from heat killed S3 strain to the R strain, and that is causing the infection. What is that? He could not tell about. Now there are three scientists called Avery, MacLeod and McCarty, these three important names. They proved that when the heat killed S3 strain was there, this particular S strain has removed, when you have what you call culture media, where you have the heat killed S3 and you will have the R2 strain. What happened when you heat kill this particular bacteria, this bacteria will try to remove its DNA out to the media. And when this particular bacteria has removed its DNA out to the media, because the bacteria will be dying, this particular DNA, which was pathogenic, was taken by R2 strain. Now when R2 strain took this particular DNA, because DNA was pathogenic, which was killing the particular, which was making some proteins, and those were killing the mice, those were becoming toxic, and those were killing the mice. So now this DNA is there in R2 strain. Now when the R2 strain will be multiplying, it will be forming some bacteria which are related to S3 type. So this combination has got, when you have mixed both of them, because of the R2 strain have taken up the DNA of S3 strain, some of the R2 bacteria have started behaving like S3. The take up of DNA from the media, the bacteria they did not combine, but what has happened, one bacteria has removed the DNA and second bacteria has taken up the DNA from the media. The method is called transformation. In conjugation you will see the bacteria will be combining. Here the bacteria are not combining. One bacteria is removing out the DNA to the media and second bacteria is taking up. The ability of second bacteria to take up the DNA from the media, it's called competency. And because of this competency, when the DNA went into this R2 strain, the recombination took place, and such a method of recombination, it's called transformation. Now these R2 strain have started behaving like S3 strain, and because of that, when the combination was giving, the mice died. So Avery, MacLeod and McCarty proved that that was DNA which was transferring from what you call heat kill S3 to R2 and making it something like S3, and then they proved that DNA is the genetic material. So the question can be asked that who did the molecular characterization for Griffith's experiment, or who did the molecular characterization for transformation experiment? Your answer will be this. The question can also be like, who proved that DNA is the genetic material? Don't answer Griffith, because Griffith could not tell about what is being transferring. Your answer would be Avery, MacLeod and McCarty only, because they have found out that this was particular DNA which was transferring, so that the DNA is genetic material. So this was all about transformation, which is one among the sexual reproduction methods in bacteria. In next session, we'll be discussing about conjugation.